So as I look over the content for this lecture, it seems like there's really not that much. So I suspect it's going to be a pretty short lecture. I'm sure you don't have a problem with that. And by the way, this is lecture 7A, and you did not miss lectures 6A, 6B, 6C, and 6D. The way the schedule was handed to me, module 6 is the exam, the first exam that we had in the semester that I'm recording this. Okay, so yeah, lecture 7 is the first lecture. Module 7 is the first module after exam 1, at least in the fall semester of 2022. Okay, I suspect I'll be using these videos in future semesters, and so that's partly why I'm always telling you, I'm emphasizing that when I give you the readings, they are for the 9th edition of Campbell's Concepts and Connections in Biology, because I suspect that really soon we're going to have to make the shift to 10th edition, which has already been out for a couple of years. We make the command decision to keep the older edition for as long as possible because it saves students some money. And when that becomes unfeasible, then we're kind of forced to move into a 10th edition. And who knows, maybe in the 10th edition, the modules, the readings are going to be in slightly different locations in the textbook. So, hey, if you're one of the students that's going to be viewing this video in a future semester, and I am not going to record these videos every semester, we're going to be making adjustments on the readings for the 10th edition, and who knows, maybe even the 11th edition, okay? I figure I'll be using these videos for 3-4 years, and then I will, at that point, record them, okay? So anyways, we don't have too much to talk about here. There's a big opening part, and let me move over to that chapter. Okay, I'm actually going to go over to this one. Right. And there's a big beginning section on different kinds of microscopy. You've got light microscopy. You've got scanning electron microscopy. This is one that actually looks like a photograph. And then you've got transmission electron microscopy. If you try this, the thing that you want to pay attention to there is the relative size. How much have these two micrographs been magnified? There's a big difference in the actual size. I mean, they look the same size, right? That paramecium basically goes from here to here. This toxoplasma goes from here to here. But if you look over at the magnification levels, this is SEM 580X. It's basically made bigger 580 times. This one has been made bigger 9,140 times. Which one of these cells is bigger? Yeah, hopefully you'll be able to figure that one out. And then there's differential interference contrast microscopy. There's a lot of variation, and so I'm not going to make a big deal out of that because I don't anticipate making that a main concern of the examination, right? What I do want to stress with you, and I think you might have seen that from my outline before, is that the size of things that are of interest to us at this juncture in the semester are really small. Okay, and so in our laboratory in the first week of the semester, we had the metric system, and we learned about meters and millimeters and centimeters, and we used a meter stick to measure things. And so we also mentioned that there are other units that are smaller than a millimeter, right? So 1 millimeter is actually equal to 1,000 micrometers, right? So this Greek symbol looks like a U. It's the Greek letter mu, right? That's the way you say it. It sounds like you're saying M-E mu, okay? You can say mu. That's the Greek letter mu. The lowercase mu is the symbol for micro, which means 1 over 1 million. 1 0.00000, right? And then when you look down the scale, looking at the size of viruses, viruses range from, actually there's some viruses that are really, really tiny. They range down to 10 nanometers, but N here is the regular English N, stands for a nanometer. And a nanometer is 1 over 1 billion. 1 over 1,000 million. And so basically when you go from a millimeter to a micrometer, that's like 1,000-fold difference. Micrometer is 1,000 times smaller than a millimeter. Go from a micrometer to a nanometer, you're basically going down another 1,000-fold. And so, yeah, a nanometer is one, there are 1,000 nanometers in a micrometer. And so as we're going down the scale here, every time we go, we make a blip, right? So from 0.1 meters to 0.01 meters to 0.001 meters, which is a millimeter. Every one of these units, these steps downward is a step downward by 10-fold. Right? And so we're really talking about things that are much, much smaller than what we can generally relate to. And so I guess, yeah, my lesson here is that cells are really small. They're really, really small. And how small are they? Well, it kind of depends on what you're talking about, because we've got two different kinds of cells. Okay, bacteria, prokaryotic. We mentioned that early on. Much smaller compared to animal and plant cells, which are eukaryotic. Okay. And that's basically the lesson here, right? Cells are really small, and the question that comes up next is, well, why are cells really small? Okay. In fact, if you look back at the whiteboard, you look back at the whiteboard, that's really all we're talking about, right? We're saying prokaryotes, bacteria, are small. They're around 1, fill this in, micrometer, using the Greek letter mu. That's the correct symbol. You can also use a lowercase u. Okay? When you see this, it basically means the same thing as a micro. So this is kind of like a surrogate mu symbol, okay? Because a lot of typesetters don't get to have mu's in their typesetting equipment, right? Next question: why are cells small? Okay, and here's maybe the biggest learning that we have in today's discussion. It's the old surface area to volume ratio argument. You'll see this SA for surface area over volume, right? That's a fraction. The ratio of surface area to volume is the thing that we talk about when it comes to understanding what are the constraints that keep cells from growing bigger than a maximal size, right? A cell can grow and grow and grow and grow and grow, but as it's growing up, its surface area to volume ratio goes down, right? When you think about it this way, if you are a very tiny cell, you've got a certain surface area to volume ratio, and you grow bigger, simply getting bigger and bigger and bigger and bigger. You might say, well, this cell over here is much bigger and it's got a larger surface area, but the surface area has not kept pace with the total volume that's inside the cell, right? And so the idea is that as the cell gets bigger in diameter, the surface area to volume ratio decreases. And well, first of all, Why does that happen? Well, that's a geometric argument. Okay, so you have to have a little bit of appreciation for geometry. And you can go back to our chapter in textbook. Let me get rid of that, and we could. I think we're done with this section of the chapter. Go to the next portion in the textbook, and you get to an explanation of the surface area to volume ratio, right? And so there's a really nice diagram of this. So I usually draw this on the board for people, but since it's nicely done for us in the textbook, no point in my doing that. We can just talk about this. And so what do you want to visualize here? Okay, is a cube, a die. And so that's what these little things are representing. So if you had a die that was 1 centimeter across one edge, right? You could say that the total surface area of this die would be, well, this side over here would be 1 centimeter square. Well, 1 centimeter times 1 centimeter would be 1 centimeter square. And we've got 6 sides of that, right? 6 sides. And so the 6-sided die would have the total surface area equal to 6 square centimeters, right? If you look at a much larger cell, a cell that is 3 centimeters across one face, 3 centimeters, then this face is going to have a total of 9 centimeters squared, right? Because that's what you get when you have 3 times 3 centimeters squared. And so 9 centimeters squared times 6 is equal to a total of 54 centimeters squared. And that's the surface area of the larger one. So yeah, the larger cell has a larger surface area, 54 square centimeters as opposed to 6 square centimeters. Okay? The volume is increasing at a much faster rate as the cell goes from a 1 centimeter cell to a 3 centimeter cell, right? Because if you look at the total volume of the first cell, it would be 1 centimeter cubed, right? 1 times 1 times 1, that would be giving me 1 centimeter cubed. And so the surface area to volume ratio, surface area divided by volume, the ratio here is going to be 6 to 1, or that's going to be equal to 6. Okay. Now, in the bigger cell, right, the volume is going to equal 3 centimeters times 3 centimeters, 3 times 3 centimeters. And that's going to give us a total of 27 centimeters cubed. Right? So this bigger cell has not only a larger surface area, it's going to also have a lot larger volume as well. And if you look at the ratio of the surface area to the volume, it will be 54 divided by 27. And that's going to equal 2, right? And so this is the calculation that we're making, right? The surface area to volume ratio is much smaller in the larger cell. Okay? Why is this significant? Well, one way you can get around that, one way you can retain lots of surface area is by having the cells increase in number, and instead of growing from one small cell into one much bigger cell, if you just simply undergo cell division, and so instead of making one large cell, if you were to make 27 small cells, right, your total volume would still be 27 27 centimeters cubed. But now what you have to say is, well, we've got 27 small cells. Each small cell has a surface area of 6 centimeters, so 6 times 27, and that's going to give us the 162 centimeters squared, which is the unit that we have right there, right? In other words, you take the surface area divided by the volume, you still have that ratio of 6, right? And so, that's a geometry-based argument that you're going to hear again and again. And the rationale here is that, cells stay small because they're able to, there's a way that they could increase in total volume without sacrificing surface area to volume ratio. And that is by keeping the cell small, just increasing the number of cells as opposed to increasing the size, right? And so what I'm trying to do here is I'm trying to answer the question, why are cells small? Answer, well, it has something, try to think about how you would answer that in your words, because this is the sort of thing that you're going to need to do when I give you an examination of the content, not just for this. I mean, maybe this would be a question that I might be asking you, but this type of question is the sort of thing that you need to be able to, articulate in your own words, right? I mean, you might want to pause the video right now, write something down, and see how it stacks up. Okay, so go ahead, pause the video. Okay, I'm going to start talking now. And so in that little explanation for why cells are small that you wrote, it should include something like, well, small cells have a higher surface area to volume ratio, and it's surface area to volume ratio must be important for cells in some way, because if it weren't important, then cells would simply get larger and larger and larger, right? I don't know if that's not a great answer, but it's the whole idea is that we're including the concept that surface area to volume ratio is important, and we're maintaining it by keeping cells as small as possible. Okay. So let's clear these drawings and move on to something else. Now, the next question in line, I think the next obvious question is, well, why? Now, why is surface area to volume ratio important? Legitimate question, and I'm so glad you asked that, right? And the answer is that if you have a cell, whether it's spherical or cubical, it doesn't matter what the shape is, the surface area is all plasma membrane. Right? It's that structure. I'm going to scroll up and scroll down. Okay, so we looked at this something like this before. In fact, I'll just leave it there. Okay, and we learned in a previous lecture how the structure of the plasma membrane is one where you've got this kind of lipidy hydrophobic middle layer. Right, that's supposed to be hydrophobic. Got lag on my sketch pad right now. And the phosphate heads are interacting with the water. This is the phosphate head on one layer. This is the phosphate head on the other layer. And inside the cell, we've got a water-based or aqueous world. Outside the cell is also aqueous. And so you've got water inside, water outside, and this hydrophobic layer, the plasma membrane, is a boundary that separates the cell from everything in the outside world, right? which means that if you have any type of exchange of material into and out of the cell, it has to occur through the plasma membrane. Okay? What do all cells need to take in? Maybe food, maybe oxygen. If it's an aerobic cell. Let's just think about food. Okay? A food molecule like, be specific here, a food molecule like glucose has to get in the cell through the plasma membrane. Right? Once the glucose gets in, it's going to feed the stuff that's on the inside. Right? Stuff on the inside is the volume. Okay? It's like the volume of the cell is the stuff that needs to be fed. The plasma membrane is the structure that is feeding the cell, right? So what I'm saying here is that we've got a ratio of organ that feeds the cell and cell that needs to be fed, right? So if you have a high surface area to volume ratio, you've got a lot of access to the nutrients that are on the outside. If you have a much larger cell, you've got a volume here of cell stuff that needs to be fed, but you have much less surface area proportionally to feed the volume that you have inside the cell, right? So again, we're looking at the plasma membrane in a different context. We focused on the structure of the plasma membrane before. We're going to focus on it again very soon. But right now you want to think of well, what is it about surface area to volume ratio that's so important to the cell that it needs to be maintained at a high level, thus making cells unable to grow very large. Okay? Okay? That's because the plasma membrane, the actual membrane that separates the cell from the entire exterior world, is kind of like the cell's mouth. It needs to be adequate in size in order to feed all the stuff that's on the inside the cell. And if you grow as a cell without increasing, if you simply grow in size, you're increasing the volume at a much faster rate than you increase the surface area, and you end up having a cell that's unable to feed itself as effectively. So I think that's kind of it. I'm going to go back to my, whiteboard and go over the items that we're really talking about. Number one, cells are really small, right? Prokaryotes, these are the two kinds of cells we care about. Prokaryotes are about roughly 1 micrometer. Actually, it should be mu-m, right? Let me erase that. I can't erase it, so I'm going to do it this way. 1 micrometer, that's the proper way to make the unit. Eukaryotes tend to be bigger. 10 micrometers to 100 micrometers is roughly the size of most eukaryotic cells. Viruses, which are not cells, are much smaller, about an order of magnitude smaller. And proteins, which are the molecules that make up things like viruses and cells, are smaller still. So we were talking about things that are way, way, way smaller than you could see even with the most powerful microscopes that are using light. Okay? If you go to electron microscopy, you can see a lot of these things, but that's not the sort of thing that you have access to in a regular laboratory. Okay. Number two, why are cells small? We totally talked about that. It has everything to do with the surface area to volume ratio argument, and that's that. And we also talked about the plasma membrane. You can review the membrane structure, which is a good thing for you to do, but think about their significance. The plasma membrane is the surface area that needs to be maintained in adequate abundance in order to allow the cell to feed itself effectively. Okay. Now, the one thing that I'm leaving you with is the chicken egg dilemma, because every time you open a chicken egg, every time if you think about a chicken egg and the inside of a chicken egg, which has got this bright orange color, yolk. The yolk is a cell, one cell. Right? And so that's an example of a cell that's ridiculously huge. Right? But there's a reason for that. Okay, we know that one cell, the egg, is going to be the starting point for the development of a whole chicken, whole baby chicken. Right? So the one cell is going to divide. It's going to divide again. It's going to divide again. And so by the time the chick is ready to be born, maybe this is our baby chick that's ready to be born. So wing, here's another wing. Here's the body. Here's a foot. Right. So as the chicken grows into this baby chick that eventually pops out of the egg, by the time it gets to this stage, it is billions of cells. Tens of billions of cells, right? It's we're basically making billions of tiny cells starting from one cell. We have to start off one cell because it's always one cell that begins the process of fertilization, right? The sperm that fertilized this egg was also a single cell, but it was also microscopic, right? And so, yeah, there are some kind of weird exceptions to the idea that cells are always small, but there's always a good reason for understanding why that happens. Right? Now, for the record, why are eggs one cell? Well, you only can contribute one cell as a female parent. Okay, one cell contains the female genome. Sperm cell contains a male genome, so it has to be one cell, right? But the egg cell has the responsibility of carrying a huge amount of nutrition. All this stuff over here is nutrition that's going to be fueling the growth of the baby chicken up until it's ready to hatch. And so because we need to put this massive quantity of nutrition into that one cell that's going to be carrying a female genome, it's going to have to be huge. Okay. So that's it. We'll be talking more about cells in the rest of this module.